Thank you, Thomas, for this kind invitation. And a big thanks to the Royal Institution for having me here to give this talk. These are unusually challenging times. And the question is whether it's appropriate to talk about science in the context of such geopolitical events. But these events have extraordinary impact now on people. And the long-term and positive changes on our societies are often driven by innovation. So my background, I'm a neurosurgeon. I look after spinal cord injury patients, partially because I'm curious, but also because the tools that I have to treat my patients are very limited. I went into research. At the moment, I have screws and rods that I can put into their spines, but there's nothing I can do to give them back the use of their hands or walking. So we need to innovate. And the thing I'm really very excited about are cell therapies. That's the concept of using a cell as a therapy. And these cell therapies can replace lost cells, but they can also interact with their environment. So you can call them intelligent drugs. The last wave of innovation, however, happened in physics at a very particular moment in time when physics transitioned from a descriptive science, so scientists were observing and recording, to a predictive science. A special moment was the introduction of calculus to describe the laws of motion. Maths gives you a lens to look into the future. We can now calculate how long it takes an apple to fall from the tree until it hits the ground. And this, together with the creation of more and more sophisticated tools, sparked waves of innovation, from telescopes to steam engines, spacecraft, and computers. And computing, again, introduced a phase shift. Since Ada Lovelace wrote the first computer script, we've built computing power that allows software to transform the world. But here's the limitation of current computing. The hardware determines the software, not vice versa. But what if the software could determine the hardware? What if you could use computer code to control the physical nature of objects? It's already happening right in front of us. It's called biology. So we need to move away from the traditional questions of biology. How does it work? How is it built? And how did it get there? To a new paradigm in biology, in which a code script is the kernel of biological theory. In this paradigm, the nucleus is the hard drive. It contains the DNA, and these genes interact with each other to create programs, so-called gene regulatory networks. The entire space of programs you could call the operating system of a cell. And at the core of this is the genetic code. This is the way that information translates into the physical reality, into protein. And here you see a wonderful depiction of the inner makings of a cell, and all these different proteins that form molecular machines. At the top, you can see the nuclear membrane with nuclear core complexes. They're very long structures. They're proteins that provide structure to the cells, and then they aggregate to form organelles. On the right, you can see an energy-producing organelle called mitochondria. But do we have the tools to program biology? Well, we started doing so about 10,000 years ago, when we started to select plants and animals with traits that are desirable. And then in the 1700s, we learned how to hybridize plants. And today, nearly all of the vegetables and grain that we consume have been somehow influenced by us. In the 1800s, we learned about the concepts of evolution and the laws of inheritance. In the 1950s, we discovered the structure of the DNA. And in the 1970s, we discovered the first restriction enzyme. That's a protein that can cut DNA in very particular sites. And that allows you to engineer biology. So we've used it to generate bacteria that create insulin. And insulin is a very important drug now that treats patients with diabetes. In the 1980s, the tools became more refined. We started to use them to create plants, modify plants. In the 1990s, we learned how to clone mammals. Some of us will remember Dolly the sheep. In the 2000s, we sequenced the human genome. 2010, we built very refined tools for engineering DNA. CRISPR, the molecular scissors that allows you to cut the DNA exactly where you want it to cut. And we've also learned how to activate programs in cells, and I'll come to that. So the tools are very sophisticated, but what about the maths? Again, sequencing and other omics technologies in the recent years have helped to translate biology into numbers. And attending today's conference here, the conference about programming cell identities, I learned that maths is really progressing as well. I think we're not far off predictive models in maths. So for 50 years, we've been engineering biology. And at the center of this was Greg Winter. So Greg Winter developed a technology that allows you to engineer antibody drugs in bacteria. And this allowed us to create a second generation of drugs. So the first generation of drugs were small molecules, like aspirin. This second generation is a lot more sophisticated. And more recently, we also started to program other microbes to produce materials, biodegradable materials, such as spider silk, that have been used in these very high-spec running shoes. So we've been programming lower species, but is it possible to also program higher species like mammals? Here we need to go back to the 1980s, the heydays of molecular biology. And you see Harald Weintraub here, who discovered a gene, a transcription factor. That's a gene that regulates other genes, which he called MyoD. And he observed that if you activate that gene, cells switch fate. They go from a skin cell into a muscle cell. That's completely contrary to the dogma of developmental biology. So developmental biology is still the predominant dogma in science. It's based on the understanding that a stem cell forms the origin of all cells in the human body. And in order to produce a neuron or a skin cell, it has to differentiate into more and more specialized cell types. Waddington proposed a model by which a marble, you can look at the picture, marble running down a valley, and it hits certain branch points where it has to choose. These are called self-fate choices. In order to ensure that not one single cell type is missing during development, these choices are based on probabilities. Stem cell biologists are studying the chemical cues that enable us to bias these self-fate choices in order to nudge cells down particular trajectories and to create particular cell types. There are two problems with this approach. Number one, development takes time. It takes about 9 months until a baby is born. And second, these cell fate choices, there are a lot more of them on the way to become a specialized cell types. So you're accumulating a lot of probability and chance events, and that causes inconsistencies. Today, long protocols with lots of inconsistencies are the major bottleneck for manufacturing cells. And it's the reason why we don't have stem cell-based therapies in our clinic yet. But Harald Weintraub proposed a completely different paradigm. If a single transcription factor can switch cell fate, we have to relearn biology. Unfortunately, Harald died 49 years old, and his work was forgotten for nearly 30 years, until Shinya Yamanaka surprised the world with a new discovery which earned him his Nobel Prize. So Yamanaka discovered that a combination of 4 transcription factors is able to program a skin cell back into a stem cell. And this stem cell behaves just like an embryonic stem cell. So this allows us to create stem cells from every individual. Each of us has their own repair kit. And the second thing it did was take away all the ethical constraints in the use of embryonic stem cells. There's no need to chop up an embryo. Finally, this is an early stage, he maybe even opened the door to cell rejuvenation. Now, Marius Wernick, who was in the audience, was inspired by this work and asked the question whether this paradigm can be generalized. So he showed that you can take a skin cell and program it into a brain cell. And then he took a stem cell and programmed into a brain cell. And then, I have no idea why you did this, Marius, you took a liver cell and turned it into a brain cell. In any ways, I think this showed that this is perhaps a generalizable principle. But there's still a problem with cell reprogramming. It's often very inefficient. This is also what we found when I started my lab at the University of Cambridge. And the objective that I had was to program human oligodendrocyte precursors. That's a specialized cell in the brain that's really important in repair processes of the spinal cord and the brain. So I was joined by Matthias Pawlowski, a very talented PhD student who now has his own lab in Germany. And within a fairly short period of time, he figured out a transcription factor combination that allowed him to produce these cells. And you can see a beautiful depiction up here. The problem was that this was a very rare event. About 1 in a million cells turned into oligodendrocytes. So the dogma was, you need to refine the transcription factor combination. The program isn't right. But his data showed something entirely different. It suggested that there's a new problem that we were facing called gene silencing. So the stem cell recognized that someone is wanting to activate a new program that is inconsistent with their current state, and they shut it down. So we decided to tackle this problem, and for a few years, we only made incremental, change, improvements. In fact, it's quite hard, actually. I used nearly all resources that we had. We put the credibility of the lab, the survival of the lab at stake. And we were quite desperate until one moment, Matthias called me and showed me the following video. So here you see human stem cells converting into brain cells within days, so 10 times faster as previously. And the incredible thing here was that every single cell in this culture turned into a neuron. And these neurons are the brain cells that connect to each other. You can see fine processes. And then they start signaling electrical impulses and reach maturities that you haven't seen before. So my first reaction is, this is not possible. He probably had photoshopped this video. Yeah, these students in Cambridge are pretty smart. So he took me down to the lab, we looked at the cultures, and it was real. So I thought, probably the stem cell that we were using was broken. So let's take another stem cell. Takes 6 months, he came back, it worked again. And then I thought, okay, this is a Maris-Wernick protocol. I mean, this is probably very unique, and therefore this is still a glitch. But over the years, our lab, company, and collaborators have been able to show that it works again and again and again for various different cell types. Now let's look at that. If you think about disease, every disease manifests at a cellular level. So every cell is potentially a treatment or a model to study this disease. So the impact of this is quite incredible. The way we achieved this was by inserting the genetic program of the cell into specialized locations in the DNA, so-called genetic safe harbor sites. And they protect the cell, but they also protect the program from silencing. This creates a control system that allows us to switch on genetic programs at will. So the cells can't escape. And this allows you to create all sorts of different cell types, like the neurons that we've just had a look at. So the next challenge here is, how do we identify the cell type programs that encode each of the cell types? Every human cell has 20,000 genes. And at any particular moment in time, approximately 10,000 are switched on. And these determine the function and the identity of a cell. Of the 20,000 genes, 2,000 genes are transcription factors. These are the genes that regulate the activity of the other genes and themselves. And what we've seen from studies emerging over the past decade, that about 1 to 6 of these transcription factors encodes a cell identity. So every cell has their own unique combination. So the question is, how do we read this out? If you look at the number of experiments that are required, this is pretty large, 8.8 times 10 to the 16th. I've calculated this, approximately 1,000 scientists working for 3,000 years. That's going to be difficult. So we had to come up with something else. And to do so, to think deeper about this problem, we entered a collaboration with the London Institute of Mathematical Sciences. So they were able to show that the way that DNA, RNA, and protein interacts in a cell creates a set of constraints that limits the possibilities and already reduces this by orders of magnitude. And we've also been able to refine the experimental approach by breaking down these pools of transcription factors into smaller pools and ways of screening them. We're now at a point where we actually can read out all cell type programs in one big effort. It's not going to be cheap, but it's doable. But imagine the impact that this could have. So this is a DARPA-style project, and I'm really keen to get this off the ground. The next question is, how is a cell type actually determined and defined in the genome? So there's Waddington's landscape again, and there are really two possibilities. On the left, you could imagine that a cell is entirely predefined in the genome, so it sits in its own little valley. And on the right is the opposite hypothesis. A cell type is a label that we attach to a combination of subprograms. So if the second were right, we should be able to combine programs from different cell types. And we again have to go back to the 1980s to Harald Weintraub's work, because he asked that question. And he showed that we can combine a cell program of a muscle cell and a pigment cell, a melanocyte. And he also was able to create some sort of hybrid of a brain cell and a muscle cell. And we've done similar things using transcription factors, combining a brain cell with an immune cell. This confirms that the operating system of cells is created by submodules. And it opens the opportunity to create cell types that actually don't exist, potentially have therapeutic use. So then, the next question that we always get asked is, how close are these reprogrammed cells to the real cells that we have in our body? Traditional stem cell biology has struggled to produce cells that resemble the cells in our body because often they're stuck in an embryonic or early immature phenotype. But if you want to do a therapy, or if you want to do research, often you'd like to have the mature, the adult cell type. So this picture up here is a landscape that we've created with data from the Human Cell Atlas. So the Human Cell Atlas is an international consortium that is trying to chart all cells in the human body. And they're using a technology called single-cell sequencing. So they can read out thousands of genes in single cells. And so if you look at the top, this is a picture, this is a landscape of liver cells, the cells that you can find in the liver. At the top, you can see adult hepatocytes, liver cells. And at the bottom, they're the fetal, the embryonic, early stage counterparts. And in the middle, there are all sorts of other cell types that you can find in the liver, from immune cells to fibroblasts, etc., etc. We can now create liver cells with transcription factors, and we can project them onto this landscape. And what you can see here, for the first time, we were able to produce fully mature hepatocytes in the top, their fetal counterpart, and some of the intermediate stages as well. So now, we've got about a technology that allows us to program biology. What are we going to do with it? I'm a medic, so my primary aim is to use this technology for medical applications. And the technology that we've created in the lab has been spun out into two companies. One of them is bit.bio. Bit.bio is the leading cell reprogramming company right now. We've been able to raise more than $200 million so far. And the objective is to create cells that we can use to democratize science, give these to scientists, but in particular to also create a new set of affordable cell therapies. We're incredibly privileged to work with some of the leading innovators. And this includes the chair of our board, Hermann Hauser. He's been at the center of two new industries. He's one of the co-founders of ARM, so chip industry, but it also sparked the sequencing industry by backing the technology that now most people are using. And I've already told you about Sir Greg Winter and Marius Wernick. I'm also extremely privileged to work with Roger Peterson, who is one of the forefathers and founders of human stem cell research and the Cambridge Stem Cell Institute. So with a consistent and scalable source of human cells, we can change the way that the drugs are developed. Let's think about Alzheimer's. We've not been very good at creating Alzheimer drugs. And one of the main reasons is because there's a difference between the animals that are used for research and the human beings that suffer from Alzheimer's. In fact, there is not a single mouse on this planet that suffers from Alzheimer's. This is an entirely human condition. So in order to create Alzheimer drugs, scientists engineer the mice so that they get something that looks like Alzheimer's. And then companies run a very efficient drug discovery process. And at the end of that, when the drug enters in the clinic, we learn that it doesn't work. In fact, we learn that probably what's been created in the mouse isn't quite what we need to treat. So the way to fix this is to use human cells, the cells that are actually affected by the condition. But what I'm most excited about are cell therapies. This is the first child that has been treated by a cell therapy. This therapy uses blood cells, immune cells from the patient and engineers them in order for it to recognize cancer. So she's been cured from a blood cancer. This is a very complicated process at the moment. It's extremely expensive. One treatment costs hundreds of thousands of pounds. Imagine we can use this software approach to create cells at scale and make cell therapies affordable. This therapy was based on T cells. They're not very good at going into solid tumors. They're rejected. There's another immune cell type that is much better suited to that, called macrophages. So here you can see macrophages that have been programmed with our technology, and they're fighting bacteria. As they eat them, they turn red. This is just the beginning. Think about combining cells in order to print organs and organoids, or using nerve cells and integrate them into bionic devices to control artificial limbs. Moving beyond medicine, the second company uses the technology to create cells for the manufacture of meat. This will not only allow us to fine-tune our steaks, but it also will tackle some of the challenges associated with farming, like the use of antibiotics, causing bacteria to become resistant, or the greenhouse gases that are associated with farming, and the risk that another virus transfers from animals to human beings, causing another pandemic. But this approach might take us beyond our planet, because I don't think it's feasible to take a cow up to the station that we're going to build on Mars. Finally, we may be able to use this technology to create new kinds of computers. Here you see a living computer chip made of brain cells that are learning to play Pong. So, when software determines the hardware, anything seems possible. Thank you.